Well, it's a pleasure to be here to try to teach a little bit about norepinephrine and its relevance to dysautonomias, whatever they are. Everything I'm going to be showing is in this book, and the book is available for free. You just have to email me, and I'll probably send it by way of Dropbox. But you don't have to take any pictures because it's all in the book. For those of you who didn't have a chance to hear the introductory lecture about the autonomic nervous system, the upshot of it is that there isn't any autonomic nervous system. That's supposed to be shocking. Instead, the autonomic nervous system has parts. It has components. There's the enteric nervous system in the gut, the parasympathetic nervous system, the sympathetic noradrenergic system, which is what we're going to be talking about, the sympathetic adrenergic system involving adrenaline, and the sympathetic cholinergic system involved with sweating mainly. So the autonomic nervous system has parts, and those parts are differentially involved in different diseases. So the concept of my autonomic nervous system is hyperactive or something like that, I don't like that. You've got to be more specific. It makes a big difference in terms of the types of symptoms, the types of signs, the types of diagnoses, the types of treatments, and the long-term outcome. Well, today I'm going to be talking about catechols, and this chemical structure is called catechol. It doesn't actually exist in the body, but catechols are important, as you'll hear shortly. And I know what the chemical structure of a catechol is because catechols look like the head of a cat. This happens to be the cat that belongs to our lab's director. And it's just this perfectly hexagonal thing. It's so cool. Anyway, you've got these two adjacent hydroxyl groups on a benzene ring, and that is what a catechol is. You'll never forget it. Now, what does a catecholamine? A catecholamine is a catechol, but it has this hydrocarbon tail at the other end. And at the end of this hydrocarbon tail, there's a nitrogen group, an amine group, like ammonia. So a catecholamine is like the entire cat, from head to tail, in its litter box. That's a catecholamine. Here are the body's three catecholamines: dopamine, norepinephrine, and epinephrine, which is synonymous with adrenaline. You can see they're all catechols, and you can see they all have that N at the end of this hydrocarbon tail. Now, the catecholamines of the body are made by a particular cascade, and it's kind of important to know what that cascade is. The starting point is tyrosine. Tyrosine is an essential amino acid. The body doesn't make it, you have to eat it. Is tyrosine a catechol? Anyone? No, it's not a catechol. It doesn't have two ears. Come on. Right? It doesn't have two ears. It only has one ear. So tyrosine is not a catechol. And it's a catechol amino acid. It's a little tricky because you've got this amine group, but then this carboxyl group. That's called an amino acid. Tyrosine gets converted by a very important enzyme called tyrosine hydroxylase to this amino acid called DOPA. Is DOPA a catechol? Who said no? That's the first time that's happened. All right, DOPA is a catechol. It's a catechol amino acid. And importantly, it's DOPA that is the single precursor of all the catecholamines, dopamine, norepinephrine, and adrenaline. DOPA gets converted to dopamine by an enzyme called, it doesn't matter what it's called, but it's DOPA decarboxylase. The carboxyl group comes off, see that? Now you're left with a catecholamine, right? That's dopamine. Because of this carboxyl group coming off, carbon dioxide is produced. And if you wanted to make neuronal soda pop, what you would do would be to take DOPA, react it with DOPA decarboxylase, that enzyme, with this cofactor, pyridoxal phosphate, vitamin B6, and you could actually see bubbles of carbon dioxide. Also, the solution would be tan, so it would be sort of like ginger ale, because catechols all spontaneously oxidize, and when they oxidize, some of the oxidation product is colored. Some of you have heard about the dopamine deficiency in Parkinson disease. This is a classic neurochemical finding. I think it was the first neurochemical abnormality defined in a neurodegenerative disease, the loss of dopamine. And to treat Parkinson's, levodopa treatment was miraculous. Dopamine doesn't get, if you could drink dopamine, nothing would happen because it gets broken down in the gut. But DOPA, the amino acid, some of it gets into the bloodstream, from the bloodstream into the brain, and once inside the brain, can be turned into dopamine, and that's why it's a great drug for Parkinson disease. But the limiting factor in terms of DOPA treatment is nausea and vomiting, because dopamine in the outside the blood-brain barrier and the vomiting center at the back of the brainstem causes vomiting. So that was the limiting symptom for DOPA. Carbidopa, which is carbidopa a catechol? Yeah. Carbidopa is a catechol, and it blocks DOPA decarboxylase. So, but it doesn't get into the brain. So it will prevent the vomiting if a patient is taking DOPA, and that's why. And meanwhile, the DOPA can get into the brain, and once inside the brain, then it can be turned into dopamine. So this was a miraculous finding. And Sinemet, anybody have any idea what Sinemet means? That's the brand name for this combination. Sinemet comes from the Latin for without vomiting. Ooh, all these light bulbs. That's kind of cool. Sinemet is still used. It's the most effective treatment for symptomatic Parkinson disease, and now you know why. And we're going to be returning to this when we get to the more modern day norepinephrine equivalent, which is droxidopa or Northera, the Lundbeck drug. Dopamine gets turned into norepinephrine by a variety of factors. This is dopamine beta hydroxylase, or DBH. Dopamine gets turned into norepinephrine by addition of this hydroxyl group here. So it's dopamine beta hydroxylase. You need this enzyme to put that hydroxyl group on there. If you have DBH deficiency, and it's a rare cause of norepinephrine deficiency, If you have DBH deficiency, you'll have all the signs and symptoms of sympathetic noradrenergic failure, but the other components of the autonomic nervous system are going to be intact. So that's a great example of a dysautonomia where there's no problem with sweating, there's no problem with GI motility, with being able to make spit, with urinary, et cetera, et cetera. All you have is severe orthostatic hypotension because of the lack of norepinephrine production. Copper is an absolutely required cofactor for DBH to work, and you'll hear later about a disease that involves a problem with copper that produces a form of dysautonomia. Probably nobody's ever heard of that, but it happens. And you need vitamin C for this process to occur. So if any of you've eaten or taken stress vitamins, stress tabs, and they all have pyridoxal phosphate, and they all have vitamin C. I don't know why, but maybe it has something to do with this. Maybe there's some sort of rationale for why stress vitamins are stress vitamins. Now, it's important to know that norepinephrine, that in the neuron, dopamine is made in the cytoplasm, the cell juice. But norepinephrine is made in vesicles, storage vesicles, little bubbles. And that's because that's where the DBH is. That means that in order to make norepinephrine, not only do you have to have these cofactors and so forth, you have to physically take dopamine up into the vesicles. And that's a very energy-requiring process. So if there's an energy crisis in the neuron, let's just say you've got some mitochondrial problem, and you can't generate ATP, the energy for this proton pump, then you're not going to be able to make norepinephrine, even though all the other aspects are intact. Now, when norepinephrine is released, by a process called exocytosis, where the vesicle fuses with the membrane surface, and then you could have this sort of omega sign. The inside of the vesicle is now open to the extracellular fluid, and the soluble contents come out, all the contents come out. So norepinephrine comes out by exocytosis. Most of the norepinephrine that comes out gets taken right back up, and it's recycled. It was the discovery of this recycling process that led to a Nobel Prize by a predecessor of mine, Julius Axelrod, in 1970. So it was important. There's another fate of the catecholamine that's of the dopamine. I said the dopamine gets taken up in the vesicles, turns into norepinephrine, exocytosis. But in addition, and the norepinephrine is recycled. So now you have catecholamine in the cytoplasm. In addition to being recycled into the vesicles, it also, these catecholamines are also broken down, catalyzed, metabolized by monoamine oxidase. This thing that looks like a scrambled egg is supposed to be a mitochondrion, because MAO is in the outer mitochondrial membrane. And these are important metabolites called dopal and dopagal. Hardly anybody knows about them. The world's literature on dopal is like 60 articles. Nevertheless, all of the neuronal metabolism of dopamine passes through dopal. It's just that dopal is further broken down by an enzyme, aldehyde dehydrogenase, to make dopac. And anyway, it gets kind of complex. It doesn't matter. It doesn't have to do with norepinephrine. The norepinephrine that gets released, a small amount of it makes it into the plasma. And that's important because we can measure it. And you'll be hearing about measurements of plasma norepinephrine levels in this conference, if you haven't already. In addition, the dopa-gal gets metabolized to a compound called DHPG, and DHPG comes out, and we can measure that. And that's really important, because if you found that norepinephrine level was high, and it is high in a lot of POTS patients when they're upright. Why is it high? Ordinarily, investigators or clinicians think, well, the norepinephrine is high, you must have high sympathetic nervous system outflow. But you can see if you had a problem with the reuptake, your norepinephrine would still be high, even though sympathetic outflow is normal. On the other hand, if you had a problem with this net, then when the norepinephrine is released, it can't be taken back up. And then DHPG won't be increased. The norepinephrine will be increased, but DHPG won't be. And that was the first, or one of the first clues biochemically that led to the discovery of the only known genetic cause of POTS, which is a genetic deficiency or a mutation of the enzyme that encodes that norepinephrine transporter. So you want to, if you're going to set up a biochemical laboratory for autonomics, you wouldn't just want to measure norepinephrine. You'd want to measure DHPG. Now, I want to mention that norepinephrine is also a chemical messenger in the brain, an important one. Exactly what it does is unknown. My guess is that norepinephrine in the brain has a lot to do with vigilance, with memory, with the encoding of distressing events in long-term memory. So I think norepinephrine has a lot to do with PTSD, for instance, but it's not really well studied. That's all I'm going to say about norepinephrine in the brain. Skip that. Okay. What conditions are associated with failure of the sympathetic noradrenergic system? Probably the most common is diabetes. Among the diseases where there's sympathetic noradrenergic system failure as part of a neurodegenerative disease, probably the most common is Parkinson's disease. But you can see there's a whole range of diseases where there's sympathetic noradrenergic system failure. Don't write this stuff down, it's in the book. What are the symptoms and signs of sympathetic noradrenergic system failure? Symptoms are things that people feel. So a doctor is not going to be able to say you have orthostatic intolerance. The patient says that. I feel dizzy, lightheaded, I feel like I'm going to faint, I get sudden fatigue, blah, 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 blah. Signs are things that are objectively measured from the outside. So a person has orthostatic intolerance from orthostatic hypotension, meaning a fall in blood pressure every time the person stands up. The orthostatic hypotension is a sign. The orthostatic intolerance is a symptom. They don't necessarily go together. POTS, for instance, you have orthostatic intolerance, and in most cases there isn't orthostatic hypotension. People with orthostatic hypotension, a lot of them don't notice that they've had a fall in blood pressure. So they have orthostatic hypotension, they don't have orthostatic intolerance. There are a variety of signs and symptoms of noradrenergic deficiency, and probably if you look down those, you'd say, you know what? That's me. Here's another way to look at the same thing. In people who have sympathetic noradrenergic system failure, the cardinal sign is orthostatic hypotension. So the fact that I'm able to stand here and give this talk means that I don't have sympathetic noradrenergic system failure. Here's what orthostatic hypotension looks like if you were going to measure somebody's blood pressure, and this indicates when a tilt table was tilted up and tilted down. And you see this progressive fall in blood pressure, and considering this fall in blood pressure, the increase in heart rate is kind of puny, and that's a clue that this patient has barrel reflex failure. You can also see these little blips that become more frequent when the person is tilted up. This is the person's heart rate, instantaneous heart rate. So if there's a premature beat, instantaneously the heart rate is high, then the next beat, then there's a pause, and so the heart rate is low. So when you see these sorts of things, gives a suggestion that there's a problem with a heart rhythm when the person was tilted up. All right. Now we're going to get into the two, and I think the two main ways to determine if somebody who has orthostatic hypotension has sympathetic noradrenergic system failure. The most important way to do this is not by tilt table testing. The most important way to do this is by measuring the beat-to-beat blood pressure when a person performs a Valsalva maneuver. Who knows what the Valsalva maneuver is? That's pretty good. Who's ever performed a Valsalva maneuver? Now, everybody raise your hand. Everybody. Everybody's had a bowel movement. Raise your hand. A Valsalva maneuver is when you strain against a resistance, like you're trying to blow up a soggy balloon, you're trying to blow into a trumpet. And there are classic phases of the blood pressure response when a person does a Valsalva maneuver. In the first phase, as soon as a person starts to blow against a resistance, there's an increase in pressure in the chest, the blood comes out, goes down the arms, the blood pressure goes up a little bit. That's a mechanical thing. It doesn't have anything to do with reflexes. But then as you continue to blow against a resistance, there's pressure in the abdomen and chest, there's less blood that makes its way to the heart. The heart pumps out less blood. The brain picks up on that immediately. Where's my blood? This causes a reflex to occur. The sympathetic noradrenergic system is turned on. Norepinephrine, the chemical messenger, is released. It binds to its receptors in the heart and blood vessels. The blood vessels tighten. And because the blood vessels tighten, the blood pressure increases. Then you relax. When you relax, it's phase 3. Blood pressure goes down momentarily. It's a mechanical thing, doesn't have anything to do with reflexes. Finally, phase 4, you're just lying there. There's no problem with getting blood to the heart. The heart gets the blood, pumps the blood into a reflexively constricted vasculature. Well, that means the blood pressure is going to overshoot. Now, this is the heart rate of the same person. You can see that when the blood pressure goes down, the brain also directs a drop in parasympathetic cholinergic system activity, vagal activity, and the heart rate goes up. In somebody who has failure of this reflex, whether it's because the brain isn't getting the information about the blood pressure, the brain doesn't care about the blood pressure because there's a brain disease, the sympathetic nerves aren't there, norepinephrine isn't released, the receptors are blocked, it doesn't matter. You get the same abnormal pattern, and it looks like this. You see in phase 2, the blood pressure just goes down, 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 down. Well, that's because the person couldn't tighten the blood vessels reflexively by the sympathetic nervous system. And in phase 4, the blood pressure slowly comes up to baseline, but it doesn't overshoot for the same reason. The person didn't tighten the blood vessels. So the two signs, these are signs, the two signs of sympathetic noradrenergic failure that you can detect by measuring beat-to-beat blood pressure with the Valsalva maneuver are the progressive fall in blood pressure in phase 2 and the slow return in blood pressure in phase 4 without an overshoot. You can see at the same time, this patient has barrel reflex failure. Even though the blood pressure is going down a lot, the heart rate is not reflexively increasing. Now, here's what you see in 3 forms of autonomic failure: Parkinson's with orthostatic hypotension, pure autonomic failure, and multiple system atrophy. These are actual records from these patients. Are those records normal? No, they're not normal, because I just said that in phase 2, you get a progressive decrease, and in phase 4, there's a slow return, there's no overshoot. But they're not, so these are positive tests. They point to a failure of the sympathetic noradrenergic system. You don't know how it's failed, but somehow there's a failure there, that reflex. But there's no specificity. You see the same abnormality in different types of autonomic failure syndromes. When norepinephrine gets into the bloodstream, it disappears in a big hurry. I already talked about the reuptake, but mainly the norepinephrine disappears because it's filtered by the kidneys. What we want to know when we measure a norepinephrine level, if it's high, we want to know there's a lot of entry of norepinephrine into the extracellular fluid, into the plasma. But because it's being removed from the plasma all the time, there's always a possibility that if the norepinephrine level is high, because the clearance from the plasma is slow. So we came up with a way to measure the rate of entry of norepinephrine into the bloodstream by infusing a tracer label, a radioactive norepinephrine. And then the idea is that the radioactive norepinephrine is going to be removed. The norepinephrine that's in the artery, the patient's own norepinephrine is going to be removed to the same extent. So if I know how much of this is going to be removed, and I know how much of this was removed, I can figure out the rate of entry into the bloodstream. It's called the norepinephrine spillover technique. And if you I don't have the data, but trust me, in POTS, a norepinephrine spillover in the heart is increased. That's increased with the person lying flat on his or her back during a cardiac catheterization. It's, to me, it points to an increase in sympathetic outflow to the heart, even when the person is at rest supine. This is a heart, and what I want to show is here's the sympathetic chain. If you remember my lecture from before, these are ganglia here, and the take-home message I want to give is that the nerves that go to the heart. She already knows this. The nerves that go to the heart go with the coronary arteries. That's how they develop. And then they kind of dive in from the outside. So if you had some sort of ablation on the inside for a heart rhythm problem, whatever, you're not going to destroy the sympathetic nerves that are going to the heart because they're coming from the outside. But if you had let's just say a myocardial infarction, a heart attack, and part of the heart was dead, then that part would have no sympathetic nerve terminals. Well, we developed a way to see the sympathetic innervation of the heart, and it looks like this. This is a cross-section kind of going across here, and this thing that looks like a horseshoe is the left ventricular muscle, the main pumping muscle of the heart. Sometimes I think of it as a McDonald's M because you can see a little bit of the right ventricular wall, but the right ventricular myocardium is like a glorified vein. It's very flabby, it moves a lot, so it doesn't show up well on the scan. Oh, there's a norepinephrine spillover. Okay, so here's what you see in POTS. There's an increase in norepinephrine spillover in POTS. It's about the same as in panic disorder. Missing stuff. What happens when somebody faints? Well, there's a big fall in blood pressure. Hello. That's called neurally mediated hypotension. And that happened in this patient right here. the blood pressure, and then it goes down. But you can see the stuff happening beforehand. This is a place where measuring plasma levels of catecholamines is important. Norepinephrine and adrenaline normally increase when a person stands up, and they increase to about the same extent. But before a patient faints, before a patient has a fall in blood pressure, adrenaline is creeping up, and norepinephrine is not creeping up as quickly. Sometimes it even goes down. This is a phenomenon I call sympathoadrenal imbalance. And as far as I can tell, it happens in everybody who's going to faint. And it happens before the person faints, as you see here. Well, it would be valuable as part of autonomic function testing to obtain blood samples for catecholamines so that you can see if this pattern is occurring. But to my knowledge, that's just not done. Another thing that's not done is to measure the forearm vascular resistance. Adrenaline relaxes blood vessels and skeletal muscle. That's the main thing it does. And so in a situation where adrenaline is being released, the blood flow in skeletal muscle is going up. The resistance to blood flow, the forearm vascular resistance, is going down. And you can see that in this patient, there's a nice mirror image. Here's the adrenaline. Here's the forearm vascular resistance. At first, it goes up. That's a normal reflexive tightening of blood vessels. But then you can see this progressive fall. And right here, the forearm vascular resistance goes below baseline. If that happens, the patient is going to faint. And as you see, that's what happened in this patient. And that's what happens in everybody who's going to faint, this sympathoadrenal imbalance. So the way I kind of conceptualize fainting is this. Ordinarily, when a person is tilted up, the blood vessels constrict. That's because of norepinephrine being released. There's no problem with blood flow to the brain. But in somebody who's going to faint, because of adrenaline and because of this sympathoadrenal imbalance, so that the sympathetic nervous system can't tighten blood vessels as a reflex, the adrenaline relaxes blood vessels in the skeletal muscle, and the blood is effectively shunted from the brain to the skeletal muscle. And you can imagine that if that happens, eventually there's going to be a critically decreased delivery of blood to the brain, and the person is going to faint. This is a Venn diagram of autonomic failure syndromes. Very difficult to distinguish multiple system atrophy from Parkinson disease with orthostatic hypotension. This is the scheme that I use to evaluate people who have orthostatic hypotension. First, orthostatic hypotension is a persistent, consistent thing. It happens every time. Very different from POTS or fainting, where it's episodic. Orthostatic hypotension is consistent. I've already talked a little bit about the causes, and I talked about how you can determine if the orthostatic hypotension is neurogenic by way of that Valsalva maneuver, the progressive fall in blood pressure in phase 2, lack of overshoot in phase 4. What I haven't talked about yet is why there's neurogenic orthostatic hypotension. And in general, there are two ways that this happens. The first is that there's a problem with the sympathetic outflow from the brain. This is what happens in multiple system atrophy. And the second is that there aren't any sympathetic nerves. And that is what happens in pure autonomic failure and in Parkinson disease with orthostatic hypotension. This is the results of cardiac sympathetic imaging that I published a long time ago. This is a patient with pure autonomic failure, and you can't see on the fluorodopamine scan, you can't see the sympathetic nerves in the heart. They're just gone. Well, that's part of the disease. That's not a big surprise. This blob over here, that's the liver. That doesn't count. This is a patient with multiple system atrophy. You see the innervation of the heart is normal, and that's what happens in most patients with MSA. It's a problem with regulation of sympathetic outflow, but the apparatus is there. In Parkinson's disease with orthostatic hypotension, it looks like PAF. The sympathetic nerves are not there, they're gone. And that applies both in the heart and also outside the heart. That's, to a large extent, why the patient has orthostatic hypotension. I've done a lot of these. What about patients with Parkinson's who don't have orthostatic hypotension? Well, some of them look like this. This is normal. This is Parkinson's with orthostatic hypotension. And a minority of these patients look like this. And I think you can see I caught them in a window in time. They're losing sympathetic nerves in the heart. They haven't lost them yet. It's always the free wall that's lost first. And that's because the sympathetic nerves develop with the coronary arteries. That's why if you have a heart transplant, you can have some regrowth of sympathetic nerves. It's always in the anteroseptal base. It never makes its way to the apex and free wall of the heart because it's only partial regrowth. In neurodegenerative diseases like Parkinson's disease, it's a dieback. When does this happen? This is a patient I reported who, for reasons I'm not going to get into, had a fluorodopamine scan that already showed there was no heart there on the scan. About 4 years later, he came back for labile blood pressure and big swings of blood pressure, and we saw, of course, he had his denervation, and he said that he was walking around like a robot, sort of like C-3PO, with this shuffling gait and his elbows bent, except C-3PO was upright and he was stooped. And he was diagnosed with Parkinson's disease. Well, in retrospect, he had already lost the sympathetic nerves in his heart 4 years previously. This is very important. People have been looking for a biomarker that will predict the movement disorder in Parkinson's. Here's another important example of the power of this cardiac sympathetic imaging. This is a patient with pure autonomic failure. This is a patient with autoimmune autonomic ganglionopathy. In this condition, it's a pan-dysautonomia. All components of the autonomic nervous system don't work because they're all depend on ganglionic transmission. And you can see here, the sympathetic innervation of the heart is intact. And this is true of all the patients with autoimmune autonomic ganglionopathy that I've studied. They all have intact sympathetic innervation, as they should, because the problem is with ganglionic transmission. It's not a neurodegenerative disease. In PAF, you see the loss of sympathetic innervation of the heart. Anybody ever hear of familial dysautonomia? Familial dysautonomia is a rare form of autonomic failure exclusively among people of Ashkenazic extraction. And I think the autonomic failure part of it is from failure of complete development of the sympathetic innervation. And you can see this very nicely by the scanning approach. You see the bottom part of the heart doesn't have nerves. And in this patient, it looks very much like PAF. But it's not a neurodegenerative disease so much as a developmental disease. There's like an arrest of development of sympathetic noradrenergic system. I talked about copper. Copper is required for DBH to work. And that means that if you have a problem with copper delivery, then there's going to be a buildup of dopamine and DOPAC and a decrease in norepinephrine and DHPG. And that's what happens in a disease called Menkes disease. And this was a very important finding because if babies with Menkes disease are diagnosed quickly enough by that abnormal pattern, and they always have that abnormal pattern, then they could be treated with copper, and some of them can have a remarkable improvement, such as this kid. He's now a rambunctious preteen. And he was diagnosed based on that abnormal neurochemical pattern showing a decrease in DBH activity, not because there was a problem with DBH, but because there was a problem with copper. I think I'll skip the Pheo thing. This is how Ldops works. Ldops is the Northera that's being sold out there. Remember, DOPA gets turned into dopamine by DOPA decarboxylase, or L-aromatic amino acid decarboxylase. Ldops gets converted to norepinephrine by the same enzyme. And so why can't Ldops be like the levodopa for Parkinson's? Ldops is for norepinephrine deficiency. It's kind of a simple mindset. And it is true that is Ldops a catechol? Yes. Nice. Yes, Ldops is a catechol. You see the head of the cat there? Okay. It would be nice if you could make norepinephrine, and that would bypass the deficiency state. And it's kind of a simple-minded idea, but that's the idea behind Northera. I don't have time to go into this stuff. I wanted to mention that post-mortem, if you look in the hearts of patients with Parkinson's disease, they really do have norepinephrine. They really do have denervation. Most of them have profound loss of norepinephrine. So Parkinson's is not just the disease of dopamine. It's not just a movement disorder. It's also a dysautonomia. There's a lack of sympathetic nerves in the heart. The last part of this, last minute or so, I just want to go, let's say everything I've said is true. What would I do? This aldehyde of dopamine, Dopal, is toxic. And so I think I could slow the neurodegenerative process that causes Parkinson's by inhibiting the production of Dopal by using an MAO inhibitor. It's a little more complex than that, but that's what I'm hoping to do. What would I measure to convince a skeptic that I really slowed the neurodegenerative process? No one has ever done that. I would take advantage of the fact that once you're caught in a window in time like this patient was with Parkinson's, you lose the sympathetic innervation of the heart very, very quickly. And that means that in somebody who's already lost the free wall, still has a septum, I could put the person on, let's just say, an MAO inhibitor, and with luck, I'll show that over time, there's a slowing of the loss of the sympathetic nerves in the heart. Well, this kind of gives you a rough flavor of what norepinephrine looks like, how it works in the body, what the symptoms and signs of norepinephrine deficiency are, and at least one idea about what we might be able to do to slow the loss of noradrenergic neurons in diseases like Parkinson's disease.
Hi. Is there any validity to measuring dopamine, norepinephrine, and epinephrine in urine?
There's a lot of dopamine, norepinephrine, and epinephrine in urine. I think epinephrine in urine is a relatively clean index of activity of the sympathetic adrenergic system. If there's a lot of adrenaline release going on, some of that is going to show up in the urine. Norepinephrine in the urine is complex. I think a lot of the norepinephrine in the urine is coming from norepinephrine made in the kidneys. For instance, in somebody who's on droxidopa, the plasma norepinephrine level is increased minimally, but the urinary norepinephrine is increased a tremendous amount. So I think that the reason for that is that a lot of the norepinephrine that's in the urine is coming from norepinephrine that's in the kidneys. Dopamine in the urine comes completely from the uptake of dopa from the circulation into proximal tubular cells of the kidneys. So dopamine in the urine has nothing to do with the autonomic nervous system or with nerves, unless you want to sort of step backwards and ask, where's plasma dopa coming from? So it's an interesting question, but each substance is kind of a topic in itself. The only thing I would rely on urinary catecholamines for would be for adrenaline, because I think that's mainly coming from the adrenal medulla.
So I know a lot of people with POTS are also on antidepressants.
Mm-hmm.
And my understanding is that you can have the serotonin reuptake inhibitor as well as the serotonin and norepinephrine reuptake inhibitors.
Mm-hmm.
So is there a better one for people with POTS? Should we go for the norepinephrine reuptake inhibitor or stay away from it?
That's a good question. It's a little complex because these drugs get into the brain. That's really where they work. And so it's not so simple as what's the effect in terms of sympathetic outflow to the heart and norepinephrine levels and so forth in the heart. There's also an issue of what's the effect in the brain on norepinephrine in the brain. Very tricky. To make it simpler, I would stick with the SSRI, adding in the net inhibition, for all I know, could actually augment the amount of norepinephrine that's being delivered to its receptors for a given amount of release. And you may not want that.
So that would be bad for everybody or just people that have the hyperadrenergic component?
I'm not sure it would be bad for everybody.
Well,
I think from a clinical point of view, it would be cleaner to be on an SSRI. If I gave this lecture next year and I asked if I showed a picture of droxidopa and I asked if it's a catechol, you think you're going to be able to answer? That's good.
I have a question. I was actually most curious about what you skipped, which was the Pheo. I was diagnosed with Pheochromocytoma by Johns Hopkins and sent to NIH.
Okay.
And it was determined that I do not have a Pheo.
You have pseudo-Pheo.
This was 10 years ago, so they didn't even, I wasn't, anyway, fast forward, I have hyperenergic POTS, but I've always assumed that it must be quite hyper to be misdiagnosed with a Pheo from a place like Hopkins. Do you have an opinion about that, or?
Yeah, don't go to Hopkins to have a Pheo diagnosed.
Not helpful, but you skipped the Pheo thing.
A pheochromocytoma is a tumor that makes catecholamines, and usually, but not always, usually in the adrenal gland, which sits there. And we found, it's already several years ago, that plasma levels of metanephrines, especially norepinephrine, is the way to go to diagnose a Pheo. Norepinephrine is made from norepinephrine that leaks from the vesicles into the cytoplasm in adrenal chromaffin cells because they contain COMT, an enzyme I didn't talk about. Neurons don't have COMT. So this leakage process is going on all the time. So plasma norepinephrine is a nice clean measure of increased synthesis of norepinephrine in adrenal chromaffin cells. So in somebody who has symptoms, which it sounds like you do, simply measuring plasma norepinephrine will rule out a Pheo. If norepinephrine is normal, that's the end of the story. You don't have to go through any more tests. A tougher question is, in somebody who looks like he or she has a Pheo and doesn't, what do you call that? And we've studied that, and at the time it was called pseudo-pheochromocytoma Yes, that's pretty original. Pseudo-pheo. But I could see how there's a big overlap with hyperadrenergic POTS. Yeah.
Hi. Can a congenital heart defect in the left coronary artery impact the development of the sympathetic nervous system in the heart? Or are they just completely not related?
I don't know what the time of embryology of the coronary artery tree is. I do know that the sympathetic nervous system is immature at birth. It continues to grow. But that's a very interesting question. I don't have an answer to that. If there's an arrest or an abnormality in development of the coronary arteries, is that associated with an arrest in the development of the nerves that go with the coronary arteries? And I don't know the answer.
I have one more question. So up there, I see that you have SAMe.
Mhm.
Is that the same?
Yeah, that's the
as-
same thing you get at Costco.
It is.
Yeah, that's
Okay.
S-adenosylmethionine, and it is the methyl donor that's required for putting this methyl group here. So is norepinephrine a catechol? No, right. Because this hydroxyl group has been replaced by an O-methyl group. The methyl group comes from S-adenosylmethionine. But S-adenosylmethionine is a methyl donor for a variety of reactions in the body, and I'm not saying that it has that anything that you ingest would have an effect on your ability to metabolize catecholamines. I really don't know.
You talked about the biomarker that you see in the movement disorder for Parkinson's. When you were talking about Parkinson's in the scan, and you showed that there was a biomarker.
I'm sorry, I can't hear you that well.
That there's a biomarker in predicting movement disorders. When you were talking about Parkinson's and
I think there's more than one.
okay, is that something that you see early on in the diagnosis of Parkinson's? Or
Oh,
later
that's a
on?
good question. I showed that picture, that sequence in that patient. How often does that happen? And I didn't get a chance to talk about that, but the sympathetic nervous system abnormality and the nigrostriatal dopamine abnormality in Parkinson's are independent of each other. They're independent. That means that in some people, like what I showed, there's a loss of sympathetic nerves that's followed by the movement disorder. But there are other people where they already have Parkinson's and they haven't had a loss of sympathetic nerves in the heart. Actually, I showed that at the very end. Well, you know, the one where I was going to be testing the hypothesis. Well, you saw that the sympathetic innervation was normal for 8 years in that patient. So, and then there was a loss. It always happens, but when it happens is independent of the loss of dopamine terminals. So I think it's a very nice biomarker to have, but it's not predictive. It's independent. They're independent, I think. All right, we've run out of time. Thanks a lot.